Okay, welcome back. The last lecture I gave, I talked about two different case studies in which the CMAP data was used. And in this lecture, I'm going to switch gears a little bit and talk about how DNA sequence affects drug response. DNA sites that control gene expression are called EQTLs. EQTL stands for Expression Quantitative Trait Loci, and it describes any DNA site, like a SNP, which Roxanne has told you about, at which variation affects the expression of some gene. Often these are in transcription factor binding regions, like in the estrogen receptor binding site. The idea is that variation at this one particular spot, called the EQTL, causes some gene to be differentially expressed, regardless of a drug treatment. So here I'm showing on the x-axis, maybe if you have AA at this one particular SNP, you get expression of or you have 10 people that have AA, you have expression of this gene that sort of looks like this and is lowish on average. But if you have AG, your 10 people have slightly higher expression, and if you have GG at this position, you have an even higher expression of that particular gene. I made up the numbers on the Y. These are not arbitrary, or these are not real units, and this is not real data. But I'm just trying to show you that the genotype at one SNP can be very, very correlated to the amount of expression of a gene at a nearby gene. So here I'm trying to show that there's sort of a relationship between this genotype and how much expression you're getting. And this is without drug treatment. This is just a basal difference. I'll try and describe a little bit how that might work. I've shown you this picture before. We know that steroids diffuse into the cell and that they bind directly to DNA and affect drug response. So here I showed you a couple days ago that the receptors bind directly to this DNA, and that causes an increase in transcription, and that causes more proteins to be made from the increased number of transcripts. But the sequence that's right here, where these two receptors bind, is really, really important. You can imagine that if this receptor needs to bind to a TAG in order to stick, if I have a CAG here instead, then that receptor doesn't bind as well or as often or stay along as long, and so I don't get as much of this increased transcription. And the part about not being necessarily related to drug response, estrogen in this case is a steroid that's in your body all the time. You can add more and get a drug response, but your cell is often doing this exact thing, is often saying, okay, here's all of the estrogen that's in my body, I'm going to express these genes, and your normal life, in your normal life, your estrogen levels fluctuate. So this pathway happens all the time, even without taking drugs, and these any changes in this binding site will affect your normal day-to-day levels of transcription and protein translation too. So here's a blow-up of that. The DNA sequence right at that receptor binding region does affect the gene expression. This is a way of describing a receptor binding region. In this case, this is the estrogen receptor region. And what this is showing is that there are some spots, like spot number 3, there are some spots, like spot number 3, where you always see a G at that spot. In spot number 5, you always see a C at that spot. And other spots, like at spot number 2, where you see either a G or a T. It's drawn like this, with these big letters showing conserved spots and the little letters showing more mutable spots, so that you know, as a researcher, when you look at this, which of these positions are absolutely critical to estrogen binding and which ones might not be. So you can imagine a SNP here. Let's say you have one person has an A and one person has a G here. They both probably bind the receptor pretty well. But if you have someone that has an A here, that might screw everything up completely, and you wouldn't get no binding and no transcription. Exactly what I'm trying to describe on this slide. If you look at position number 4, a T might make a better estrogen receptor binding site than a G, even though both occur. And more binding might mean a bigger drug response for that target gene. Okay, here's a paper that describes how SNPs that are associated with drug response are likely to be EQTLs. So the SNPs that are correlated most with how sensitive or unsensitive you are to a drug are likely to affect gene expression changes. This is from Nancy Cox and Eileen Dolan. They're at the University of Chicago. And the paper is called Chemotherapeutic Drug Susceptibility Associated SNPs are Enriched in Expression Quantitative Trait Loci. So in this paper, they measured SNPs and drug susceptibility in cell lines. They grew cell lines from HapMap, CEU population in culture, so again in plastic. HapMap, I hope has been described to you. It's basically a big project to collect cells from lots of different people all over the world. So CEU is a European population of people. These are different than the NCI 60 lines because they're healthy. These are not people that are sick in any way. So we're not looking at cancer cells now. We're just looking at healthy people in different populations. And in this experiment, they added 6 different chemotherapy drugs to these cells, measured the measured how much drug they needed to kill each of the cell lines. They measured the genotypes, so the 500,000 different SNPs that differ among the cell lines. And they measured, without drugs, the gene expression of each line. So different people from this population have slightly different basal level gene expressions. They found SNPs that significantly associate with drug sensitivity. So if you're one guy, you might have a CC at this position, and that makes you slightly more sensitive to this drug cisplatin. Whereas if you're the painter dude, you have 2 Ts at this position, and you're less sensitive to cisplatin. So once they found all of these SNPs that did correlate with drug sensitivity, they classified the SNPs based on where they're located in the genome. So they basically have 3 main categories in which a SNP can fall. One is whether the SNP is in a coding region, so this is exons. In a non-coding region, this is things like introns or UTRs, untranslated regions, which are known to be regulatory, but that don't necessarily get translated exactly into the protein, but have some regulatory function probably. And EQTLs, these sites that are known to be associated with changes in gene expression. Then they further classify those 3 categories into 7 different categories. These 3 are types of mutations that cause varying levels of protein misfolding or misassembly. A missense mutation means you still get a protein, but maybe it doesn't work quite as well. A nonsense mutation means you get a really totally wonky protein that doesn't, clouds are my wild type protein. So a missense means you get a little cloud, but it has a little doohickey on it maybe. A nonsense means you don't get a cloud at all. And a frame shift usually means that you don't get a cloud at all too. In the non-coding regions, you have mutations that can occur in a 3' UTR or in a splice site. Both of these could affect regulation of translation, but it aren't going to cause the protein to necessarily misfold. And then these EQTLs, you have 2 types. One is a an EQTL, a SNP, that's exactly on the same chromosome as a gene that it controls, or that the genotype is associated with the expression of this gene on exactly the same chromosome. But you could have an EQTL that was on a different chromosome than the gene that it is associated with, and that's called a trans EQTL. So, with all of these classes, you expect that SNPs that are associated with drug response are going to be in exonic regions, are going to make your cloud turn into, are going to cause really, really big changes in proteins. So you can imagine that if you're taking this drug, a platinum drug here, and it has to go into your cell this way and go through all of this this whole pathway to affect your cell. If this thing right here totally doesn't work, then that would really affect the sensitivity of your body to the cell, because all of a sudden you can't pump out the drug from the cell as much and it hangs out more. So if you turn this guy who used to look like a cloud into something totally wonky, then you're going to have a big change in phenotype. And that's what they expected to see. But they didn't. SNPs associated with drug response weren't preferentially found in the exonic regions. So no more than random SNPs were they found in exons. So it wasn't that that for changes in drug sensitivity, the clouds weren't turning to squiggly lines. That didn't seem to be the mechanism of action. There was no enrichment for drug response SNPs in exons. So they're like, all right, well, that's weird. Keep looking, I guess. And they found that the SNPs associated with drug response weren't found in the untranslated regions, or the untranslated regions that were right next to the gene, part of the mRNA. That's not where they were. The SNPs associated with drug response aren't found in the splice sites, which is the DNA spot where introns meet exons, which is sometimes very important to decide for the cell to decide which kind of a transcript to make. So this is a splice site where this is an exon and this part's an intron. If you screw that up, maybe you accidentally skip the exon, or yeah, that's most likely answer. You could imagine that if you had a SNP right there and you messed up this machinery, then when you're making the mRNA, instead of getting boom, 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 you get boom, boom, and you're missing this whole portion. That would be bad for the protein, but that's not where the SNPs were. They weren't enriched there either. What they found was that most of the associated SNPs were either in introns, the lines between the exons, or in intergenic regions. And they wondered how variation in intergenic regions affected cellular phenotypes. So they compared these SNPs, all the SNPs they found to be associated with drug response, to gene expression. This is the same plot, made-up plot I showed you before, showing that one SNP, a genotype at one position at a SNP, can be correlated to gene expression. So they tested all of these positions that they knew to be correlated with drug response and looked for correlation with gene expression of any gene. What they found is that SNPs associated with drug response are likely to be SNPs that control gene expression, are likely to be these EQTLs. The figure in their paper, I think, is a little bit tough to understand. Basically, they were trying to show that the drug SNPs are EQTLs is a significant finding. So to make a statistical claim about that, they chose a bunch of random SNPs and counted the number of EQTL SNPs that you find, and then did that 1,000 times to get a histogram. So the in this case, if you choose a bunch of SNPs and count EQTLs, you usually get about, let's look, 120 SNPs that are EQTLs. And if you do it a bunch of times, you get sort of a background distribution of how likely a SNP is to be an EQTL. But for this drug, carboplatin, the SNPs that affect its response are way more likely to be EQTLs. So they're way out here on the x-axis. So this is just a way of showing visually that compared to a random selection of SNPs with drug response, the SNPs with the drug response association are almost all EQTLs. They did this for all 6 of the drugs that they tested. So carboplatin, which I just showed you, is here. Cisplatin, which is another platinum-based agent, is out here. And the other ones show basically the same thing. So the next question they asked was whether these SNPs are controlling expression of more than one gene or just one gene. So the definition of an EQTL is a genotype that's associated with the expression of a gene, but they thought that maybe some of these SNPs might be associated with the expression of a lot of genes. So they defined this term called the master regulator, which is a genotype that is associated with expression of at least 10 genes. So one particular SNP that's associated with lots of different gene expression changes. So here's one example of that. In the middle here is a SNP. This is an RSID, which is just a way to label a SNP. And then out here on the edges, these are all genes. And what they're showing in this figure is that this particular SNP has associations with expression of lots of different genes. So this is a master regulator, according to their definition. They make more plots that show that the drug SNPs they found are more likely to be these master regulators. This is basically the same type of thing. They sample, count the number of master regulators, do that a bunch of times, and then put one plot showing one point out here showing where the observed number is. So in all of these cases, except maybe Iraq C, the number of SNPs that are master regulators is much higher for drug-related SNPs than it is for randomly selected SNPs. Luckily for us, they made a database of all of this. It's called the SCAN database. It's available on the internet, scandb.org, that annotates every SNP. It's a physically based annotation so SNPs are categorized according to their position relative to genes, and it tells you exactly which genes the SNP acts as an EQTL for. If you're doing an experiment where you're following up a hit that you got from a GWAS, this is a great tool to use because it says, all right, well that SNP is interesting, and here's maybe an explanation why. That SNP affects the expression of these 5 genes or these 8 genes or whatever. So this is a really good resource for anyone who's doing anything having to do with genotypes and SNPs. Okay, here's another paper from the same group, Eileen Dolan, University of Chicago, showing that an EQTL-based approach identifies these 2 genes, these are genes, CTTN and ZMAT3, as markers for susceptibility to this drug, pemetrexate. So this is a drug. The experimental setup in this paper is very, very similar to the one I just described. They grew cell lines from HapMap in culture. They added this particular chemotherapy drug, which I have trouble saying, so I will just circle, and collected 3 different data sets: genotypes, gene expression of each of the cell lines without the drug, and some measurement of the drug response. So they know exactly which cell lines are responsive to the drug and which ones aren't. First, they found SNPs that are correlated with drug response. So they take all the people that are really susceptible to the drug and all the people that are really unsusceptible to the drug, and they look at each SNP to see if there's some enrichment for a particular genotype in the susceptible people compared to the unsusceptible people. In this case, they were super interested in the adverse effects of the drug. I haven't had cancer, but apparently cancer drugs are not very fun to be on, and this is one of the worst. So they were hoping to figure out if they could figure out SNPs that made you more or less susceptible to the adverse effects of the drug. Adverse events are also caused by expression changes or also correlated with expression changes. So you can think about adverse events and the presence of adverse events as being kind of a different phenotype than the effectiveness of the drug, but one that is also measurable in kind of the same way. They found, these are the same plots as they showed last time, same type of plots. They found that drug-associated SNPs that are associated in this case with the adverse event, are often EQTLs, more often than random, much more often than random, which is great. And that the drug-associated SNPs are more often than random master regulators. This is the random again. The best SNP hits are EQTLs for these two genes, CTTN and ZMAT3. And they show that the higher expression of these two specific genes correlates with cell survival. So here on the x-axis for each of these, we'll start here, you have expression of the gene. So this is expression of CTTN. This is high expression of that gene, low expression of that gene. And then on the y-axis, you have cellular survival. So things that are up here are very resistant to death with this drug, and things that are down here are more susceptible to dying. Each of these dots represents one patient. So a patient has, or one person in this population, they have some measure of expression of this particular gene, and we've measured their cellular survival. So that gives you a point. And they're saying that there's a correlation here, that this line is not, does not have a slope equal to 0. And they do that for, in two different populations. CEU is one of the populations, and 52CEF is another population with these two genes that they thought might be related, ZMAT3 and CTTN. And in all of the cases, it looks like there is a trend between the expression and the cellular survival on the y-axis. They claim that the genotype of this EQTL SNP affects gene expression, and the gene expression of these CTTN and ZMAT3 genes controls the drug response. Then they went on to do some real biology. Here they knocked down the expression of the genes. So you can basically, if you have a specific gene in mind, in this case CTTN, you can make that gene, you can silence that gene a little bit using a particular technique that makes it just less expressed. And what they're showing here is that if you take some of the drug, a little tiny bit of the drug, and you add it to the cells, then the cells don't die. And if we follow the open boxes here, that's just water. So if you add more of the drug, the cells die at a higher rate. So increasing drug on the x-axis and survival on the y-axis. So lots of the drug, 10 micromolar of the drug kills most of the cells. None of the drug kills none of the cells, and actually you get a little bump. So what they're showing, let me erase this, is that if you just add water to these cells, you get a trend that looks like this. I'll try and highlight this for you. But if you can knock down the expression of this gene, if this gene goes down, then you get less cell survival at the same conditions. So that's these guys down here. And by doing this, they show this is a very compelling argument for this gene being involved in the drug response, because if you knock it down, you change the drug response. This open circle thing here is a control for the method. So it's it should be pretty similar to the open squares, but it's a slightly tighter control than just adding water. On the other side here, let me change colors. They did the same experiment with the other gene they're interested in, ZMAT3. They show that the percent survival is lower across the board at different concentrations of the drug when you knock down that particular gene compared to controls. So the take-home messages here are that SNPs that are associated with drug response are likely to be associated with gene expression. That is, they are EQTLs. They're not likely to be missense mutations, so they're not as likely to be something that causes our functioning protein to turn into another protein. It's a little bit more fine-grained than that. It's all about a change in expression. It's not so much these huge, gross changes that we expected to find. This is actually true for disease-associated SNPs too, not just drug response SNPs. And really, I think, highlights the fact that expression is important. How much the cell is making of a particular quantity is really, really important, because that varies between people much more than the number of ways we can screw up our clouds into other things. You can imagine that the cell can only mess up a few of these clouds before it really dies, whereas it could make, one person can have 3 clouds and another totally healthy person could have 4 clouds, and those changes are a little bit more permissible to the body and are still interesting and still affect our disease states and still affect our response to drugs. So that's the big point of the whole expression study material.